Is it running? Okay, another one. Sorry for this complicated start. So we want to predict the effect of variance. No, it doesn't react to the remote. In fact, it seems to be advancing automatically. That may be, I may still have some surprises of complication in store. All right, so we want to predict the effect of a single variant upon the function of a protein. We know this is the figure for function, but I showed you somehow similar figures for structure. This is for structure. I showed you somehow similar figures for function as well. Number of residues aligned, percentage sequence identity. If two proteins have 100 residues aligned, and of those 33 are identical, essentially, if they're naturally evolved, they have similar structure. Conversely, that means you can change 67 residues in a protein. And now we're trying to predict the effect of a single variant. This image may suggest to you that most changes are neutral. That's not quite true. You understand why? Because this image is an image of a long selection of neutral changes. The neutral change is the least likely, or is not very likely to happen. But what you observe are only things that don't matter or bring a benefit. But ultimately, overall, most of what you observe really are things that don't matter, that don't change, that don't impact the way the protein functions. If it would impact it, the protein wouldn't survive, or the organism wouldn't survive, all right? So that's essentially why you see evolution is a selection, a sandbox of selection for neutral changes. And that is essentially the blue here. Now, if you have a variant or sequence variant that relates to a disease, this is typically not the kind of situation we have. So that's why we in fact want to have methods that really would detect if a single variant changes the function of a protein or the structure of a protein. There are different types of changes that can happen. But let's get one step back. The way genetic information is packaged. It's packaged in chromosomes, of which we have somehow, will that disappear?
If you go on blue, and then on the very last,
the full screen mode?
Then it's done.
Okay. Okay. And then I don't use the mouse for security. Of which we have 23 in human. These chromosomes are wrapped up like that, and inside of the chromosomes you have then, for instance, genes and other structure. Oh, you can even use the mouse now. So when you look again at the level of the genome here, you have a lot of let's call it structure, a lot of coding, a lot of information contained in the code. At some point you have the part that people refer, let's not talk about what it's referred to, where the sort of gene starts and ends, where you have the exons here, these the three red regions here, that are coming together to be the protein in the end. Okay? So before that here, you have regions that decide on how strong the gene will be expressed, how the protein will be expressed, how all of this translation of the DNA to the protein will happen. That information may be up here. All right? Now, variant means you can hit somehow this information content. You can hit things that are not directly related to how that is expressed. You can hit things that are essentially inside of a protein, one particular of these here. You can hit things that will change the way they're put together. So there's a variety of different variants. On a more simplified way of looking at it, where you have a part that is non-coding in the DNA and a part that is coding, that becomes the protein ultimately, then now a variant can be such, here you can have the A to a G in a non-coding region. In the final protein, that will still be the same. because it's in the part that is not regulating, that is not, then you can have a change here. GG in this particular variant is to the G changes to an A. That in fact also doesn't change the protein, because this one is a non-synonymous. This is a silent mutation. It's one that does not change the amino acid, because the triplets of DNA correspond to amino acids. 3 letters in the nucleotide sequence correspond to 1 letter in the amino acid sequence, and 3 times 4 is 4 by the power of 3 is 64. We have only 20 amino acids, so you have a redundant code. Okay, and the redundant code means that in fact both these triplets, GAA and GAG, code for the same amino acid. That's why in fact here the same amino acid comes out. And there can simply be the variant that would change that particular amino acid. In this particular case here, in the variant that I made up here, we in fact flip even the biophysical feature of the amino acid. So in a more simplified way, where you have a single letter on the genetic code that is changed, you have already very different possible outcomes. Okay, now in terms of classifying how important they are for risk, and now when we talk about risk, then typically this is health related. So risk in the sense of the influence upon human disease, then somehow you see that most of these things that happen outside of coding regions are not as risky. Typically, it begins to be much more risky when you change the protein sequence. Splicing regulation, that means simply how you put the protein together. This one here simply means you go into the start, you hit on the start signal. So ultimately you don't have the protein. That's why this is just more risk, because you completely delete a copy. Anyone where you can delete a copy is likely to have an impact, while these essentially change 1 amino acid in a copy that may have an impact. But not having the thing will have understandably a higher impact. When you look now at the distribution of chromosomes here, those are the 23 chromosomes, and non-synonymous in green, synonymous in red, you see that in fact synonymous variants, synonymous are the ones that do not change the amino acid, and this is the subset in the coding region, are surprisingly often. So 20 and 64, you would expect the number smaller than half, and roughly green and red, in fact, if anything are of similar height, if anything, green is actually higher than red, while 20 and 64 is less than half. So the non-synonymous in principle should be observed more often. They're not. Now, they are also important in the non-synonymous, as I said, are relevant for many of the diseases. Now let's begin with the way you can predict the effect of a variant. So I said that the evolutionary track record that we have is a track record of the evolution of total neutrality. So what survives is neutral. So that implies, and this is in fact the idea of the first method here, SIFT, whenever you look at an alignment, a family alignment, so you look at this one particular protein, you put it into its family, so you find proteins that are related to it. At the position at which you have the variant here, you then ask, how conserved is that position in that family? Or you ask more particularly, if that were to be mutated from an E to a K, in that family here, do I ever observe that E to K transition? If so, then maybe that is something that is tolerable. If not, then it's less tolerable. And that's the idea here. As simple as that. Now, what else do you need to do if you come up with this idea? What are the free parameters that you have in the game? The idea is clear. How do you transfer that idea or translate that idea into a code? So if somebody gives you the alignment and you have the mutation of the E to K, what's the only sort of unknown that you still have? And the simplest way of putting this idea into a solution.
How much similar they have to be?
So that essentially means how many do you include in this family? Okay, I'm telling you, so the condition in this particular case is I give you the alignment. So I already answered that question to you. What else is missing? So I give you a set of proteins that are related, I claim. In principle, you're right. You would have to answer, you would have to, when you run the search algorithm for finding a family, you would have to know when you to stop. Let's assume that I did that for you, yeah?
Through probabilities? Which amino acid should be in position? Which amino acid is probable to be found in this position or not?
How do you get that?
With the method from the last Thursday.
Last Thursday. Which method was that?
It was a PSSM.
Yes. But in principle, that's what I give you. You have a PSSM.
Right.
So essentially, you have, for this particular E here, you have at that position, okay, my slide is not very good. You can't see that E here, but say that is obviously, is this blue here, right? You can clearly see that. And now in this blue, for every single of the 20 amino acids, you can count how many you have in that alignment. That gives you a probability of observing the K in that family. Now what?
Well, maybe this is still related to organisms. Which organisms do I include? Which ones?
You are totally right, and all of these are important issues. But assume I give you the alignment. What else remains to be done?
What I think it's what Christian already referred to was the threshold at which
what?
At which you decide it has an tolerant or an intolerant output.
Did you say that?
Yes, he said.
I don't know.
Okay, I'm not really sure. I thought I heard him say the threshold for picking up sequences, and he dwelt on the same motif in some sense.
I think I heard that.
But anyway, that's exactly the threshold we need. Right. So we have a certain probability. Okay, the simplest one would be I transform this into a probability and I say more than half is effect, less than half is not. That would be one way. But you need some data to in fact see, or it would be more comfortable if you had some data for which you would see whether that, if you implemented that, it would essentially give you interesting predictions, or mostly right predictions. Okay, that's one parameter, right? Christian is right. So there are additional issues here. How you construct the alignment in this particular case, in fact, is slightly different from the way you do it usually. Okay, polyphen. Polymorphism phenotyping, here are the two publications of that, and the big difference here between these two publications in between, this essentially is a similar approach. Here we get into something that is essentially uses machine learning and a lot of other ideas. The original idea really was, okay, the way I described it so far, you get a simple probability and you have a simple threshold. Now this simple probability is not dependent upon, and that is essentially the way I understood Christian, you just put a threshold, you say everything here is included, and then you compile a probability. Now in this probability that you compile, you essentially count. If everything you align is sequence identical throughout, then it would appear that the E here is totally conserved, although you actually have no counts. You have only one protein, right? One way of handling that is you could sort of filter out things that are too similar. You see the problem I'm talking about? Okay, I tell you here I give you 50 sequences. Okay? Now what if I gave you 50 sequences that are identical to each other? That suggests to the method that this E is totally conserved. Not because there's any biological reason, because I simply gave you only 50. Okay? So what's the difference between 50 different ones, in which in fact if the E is conserved in 50 proteins that are very different, or if the E is conserved in 50 proteins that are very similar, the information content of the surprise, if you now have the E to K, is a very different story. It's a very different information content. How can you measure that? And that's the idea behind polyphen. So you compile a probability that considers also the overall similarity. Essentially a weight, okay? To weight the conservation, the probability of the position by the overall conservation. The hack solution could have been to sort of filter out things that are too similar. But the way this was done here is much cleaner, much more rigorous, and in fact it performed much better. Later we go into machine learning, and then we essentially go into what I will describe later.
It's not very collaborative today.
Well, the whole thing started with me throwing away my talk. That was my mistake. Okay. And now what do I do? Okay, now SNP-S 3D from the Molt group, and again this is an old publication. There is a much improved method more recently published. Here the idea essentially is, okay, so far what I looked at only is the conservation. It's done by SIFT and polyphen in two different ways of compiling the probability, but fairly simple. Now, when I look at a protein structure, whether or not I'm conserved in the inside or the outside matters. Whether I am in a conserved loop outside or in a conserved helix matters. So if I additionally used structural information, I could compile some energy. If I looked at whether this E here is contacting somewhere, is it in a high contact density? Is that high contact density inside or outside? All of that would be important. Given its alignment, would add additional information. And that is the idea behind SNP-S 3D. Once you have a structure, use that structural information, and you can compile energy levels, and these energy levels help you to sort of put the, originally just use these energy levels, the more recent version, to put this probability again into context of the structure. Now essentially, originally what we had with these were three different methods. That originally this was a simple compiled energy. Were very simple, in the sense they had a very simple threshold and they used very simple principles, evolution and structure. And originally there was no combination. Now we go into a new concept in some sense. It happened before the other one, because this was developed here in the Blundell group by Topham, and there the idea is to look at the thermodynamic cycle. So essentially there is wild type, a mutant, so you have a transition of a wild type that is unfolded to a wild type, the mutant that is unfolded, and a mutant that is folded, and a wild type that is folded. That gives you a thermodynamic cycle. And this thermodynamic cycle, you can try to gauge the impact of a single change upon that cycle. That gauge, or that impact upon the cycle, would essentially measure the energy upon the impact of a mutant upon the energy upon binding, and it would originate from statistical potentials. You simply observe how often you have these transitions. What this one here shows is the binding energy that is calculated and the binding energy that is observed for one particular protein. I can't remember which one it is. And you see there is a relatively good fit. I mean, as far as computational biology is concerned, this is an extraordinarily good fit. Okay? And it's a similar idea. In this particular idea, the experimental measure here is some particular binding. So now what we're calculating in principle is the effect upon some particular binding event. That may not capture everything. And it may not be giving you the same prediction as an alignment. In fact, it will not. Now, once you have gone to that step here, you have these data, you can go to the next step and you can go essentially and compile the changes of binding energy learned through machine learning. This is from Marianne Ruman here. And here's a similar idea where again is energy predicted through an SVM. Here we get into a concept from Weizmann that uses a similar idea, again the energy experimental delta G, predicted delta G. The difference now is we are looking explicitly at three-dimensionally defined or confined binding sites. This is essentially what we talked about the hotspots in some sense. And again, there's a fairly good fit. Now here we get to a new idea that comes from the Dick Charge group. And this is really the one that is on the web. Okay. The Lichtcharge group has this idea that essentially we have an equation that relates the change of phenotype through this equation to a particular variant. And by looking at the landscape of evolutionary change, you fit some of these terms, you get an estimate of the relative probability, and then you can actually compute from an alignment, again, the impact of change. Now this is in some sense like we went from SIFT, where we have a simple threshold for probability, to polyphen, where the probability considers the overall alignment. Here it's done in some sense bigger way. So here it is done by, again, in a polyphen kind of way, where we look at the entire alignment, where we look at the structure in the tree, what is important in the tree, so in the alignment event of things that are more related, so some clustering underneath, and you put that into a dynamic landscape. And I believe I'll leave it here, and let's get into more detail. If you want to do machine learning on this task, the problem that you have is the experimental data. So in the last 3 slides I showed you delta delta G measurements. So delta delta G measurements, essentially you have one protein, you have another protein, they bind, and then you essentially measure how strong this binding is and how that is affected by the variant. So if you change a particular amino acid, is the binding changing? Is the question you answer. In this particular case here, you have a particular variant that changes a phenotypical outcome. So a strong effect on binding, this clearly is a strong effect. You cannot put that into a number. So that's the delta delta G is a value. Here it's a binary. So these mice look different, full stop. Do they look more different than if one of them would have been gray? Or if one of them would have had half hair? Well, that would be less different. But again, so you see my point. Here, again, what the slide is supposed to point out is if I want to apply machine learning, then essentially what I have to do is create data on different levels. I could develop a machine learning device for this. I could develop a machine learning device for this. But ideally, machine learning gives me the power to somehow integrate all these different levels of data. And hopefully, the machine would learn effect as everything. On the level of disease, this is not a disease, but, well, yeah, as a human, this would be classified as a disease, I guess. Then we have to collect a data set, and one data set here is like this, that comes from the PMD. PMD was a database that collected data of impact of variance on very different levels. On the level of the molecular binding energy and on the level of the organism. Okay? It was an integration of all of these levels. One particular data set that Jana Bromberg collected in our group was separated in this way. So we had about 40,000 effect, 14,000 neutral. We got to this classification. My first question is, why do you believe that the effect dominates? Dominates quite clearly, right? It's a fourth, roughly. No idea? So most of these things measure an effect of a variant. First of all, why do you believe people measure these effects of variance? Or why do you believe? So this particular one here, you may see, is obesity. It's related to obesity. How do you believe they find it? Why does that appear in the record?
Because it's obvious that they have a problem.
Exactly. And for people where it's not obvious that they have a problem, very few people study them. Very few people experimentally, very few clinics study people who obviously have no problem. Because people with obviously no problem typically don't walk into these clinics. I mean, maybe you could call the personnel. From now on, we will only turn to ourselves or something like that. So the first bias really is selection. Most of the things studied are things that are somehow have an impact. That's the first statement. The next statement is that, again, if this mouse here would have sort of slightly different hair, it may be very subtle for you to find that out. Effect is a strong effect. Now, no effect. Well, if the mouse has patches, is that no effect? Or if I don't, in fact, see this dramatic transition, does it mean there is no effect? Here, in some sense, we can, in the methodology of binding, no effect is more easy to measure. On a larger level, typically, if I don't observe it, I don't record it. I see an effect, that is a reality. I don't see an effect, maybe I made a mistake in my experiment. So no effect is something that is much more difficult to get the courage to publish. First of all, in science, negative data is not published. There are very few ways in which you can publish negative data, and no effect is in some sense, negative data. You could turn that around and say whatever is neutral is important for designing proteins. In that sense, is positive data. It's something that I will need. But again, it's not easy to have a forum for publication. That's one problem. The other problem really is, and briefly, this came from you, Dimitri, or from you, Christian, on Tuesday, the issue of balancing out. Not on Tuesday, on last Thursday. Balancing out predictions. So if you begin machine learning with a data set that is so one-sided, loop-sided, so 1/4 are neutral, then the machine learning device is very likely to learn the same bias. That brings the next question. This would be okay if that would be the natural bias, if that would in fact be what will happen tomorrow. But that's unlikely. When you want to discover a new disease, it's very unlikely that most of the things you see are disease-related. Discovery means most of the things you see are not. Discovery means you find the exception in a big pool. That's why it's a good discovery. If you would get them out like this, and then that's a discovery, it's not a discovery, right? Now, if that is an exception, then in some sense, you expect that the ratio for your application, for what you want to do, will be inverted. So clearly, many of those that you will look at will be neutral. And you're looking for the few strong effects in some sense, right? So that means that you have to be really worrying about this distribution. And that, for instance, means that you have to try to get more variants that are neutral. Now you have to get more variants that are neutral without getting them from experimentalists. So you have to create them in the computer in some sense. You have to find a way to, let's call it, invent neutrals. Do you remember any idea how you could come up with an in silico ways to create neutrals?
The parts of the genome that don't code?
That would be a great idea. But the problem is, if you now want to predict the effect of an amino acid change on a coding region, and for the negatives you use something that is a completely different data set, then again you will most likely bias your machine learning device towards something that you will not see in the future. Possibly that machine learning device in fact will distinguish between coding and non-coding, rather than between effect and not effect. So we have to be careful that the negatives we create are negatives from a sample from a data set that we will in fact use in the future. Okay? So non-coding is a safe, surefire way, but unfortunately it brings us into a new space. So we need another idea.
Oversampling with introducing some noise?
That is certainly true. In fact, this is the simplest way. You oversample by a factor of 4 or 3, depending on or wherever you want to go. But may even be by a factor of 10. The problem is you still don't really have a lot of negatives. The problem of that approach, typically the power of your machine learning is always defined by the smallest class if you have a binary classification task. So the binary red and green classification task here, the size of this, the effective size of this data set here, will determine how powerful your machine learning device is. Okay, I said that there's a problem in the way experimental methods are biased because they look at certain places, and there's in fact a problem of really making sure that a neutral is a neutral. So we expect that this data set on top has problems. So we have a very small set, and by duplicating it 10 times or whatever number of times, I do not increase the effective information contained in it, okay? So I blow it up, a data set that I'm not entirely sure is right, and I blow it up now and I make that the dominant part of my prediction. That could be possible, and that could work. But at least you admit that this is a daring enterprise. So is there any other way in which we could do it? So, but let's get back before I make one point again. In a particular case like that, you will always, so you're totally right, Dimitri, you will always do that. But it's not enough in this particular case. You will always do that as long as you know that the true distribution is not 1/4 of the green. Okay? In this particular case, we actually don't quite know how the true distribution will be, but we are very confident that it will not be 1/4. So we will clearly blow it up. But again, this is not enough. Any idea how we can? Yeah.
We can look at the function of enzymes. And,
because I discussed that already, or?
We could have read the paper.
Mini. That is cheating. Okay, so here's the idea. You look at two proteins. In this particular case, they're both enzymes, 1 and 2. And these two are such that for both I've experimentally measured their function. And I know that their function is identical, experimentally. Both are enzymes, and they both do the same thing. Ideally, I would actually even know where they do that, where I have the binding site. Unfortunately, I don't have enough data for that. For pairs of enzymes for which I have experimental data that are not too different, similar enough in sequence, and both of them experimental information, of those pairs I have a lot. And then I simply say wherever they change, here, the blue and green, blue and red, wherever they change, this is neutral. Because these two things do the same. If these two enzymes would be too far apart in sequence, I may make alignment mistakes. And then many of the things that I find that are different may really be different. I mean, may really have an effect, but I don't capture this effect simply because I align them incorrectly. But as long as the alignment is right, so as long as these two sequences are similar enough, I'm confident that I can align them very well. I know that changes are irrelevant, okay? Essentially. Or let's call it a good guess. In this particular case, when we start there, this immediately gave us a data set that brought it all up to some equal numbers. So we had 50-50. Roughly, neutrals and 50-50 effect. Now we get into the issue of predicting through machine learning, and the first question is the machine learning device. In this particular case, we used a neural network. We simply used the neural network because there's noisy data, and they are good in an environment of noisy data. Again, the input here is the sequence, and then if I had an input window of 13 consecutive residues, I need another set of units here for the change. Okay. The central P here is, in this particular case, for instance, changed to Y. Don't ask me why. And the output is either it has an effect or it doesn't have an effect. Now, the unique... Okay, let's hope that it comes on the next slide. I know the types of features that I want to use relate to features of the amino acids, relate to the evolutionary information as we had it, relate to whether or not I'm in the inside or outside, whether I'm binding to other sites, and what type of secondary structure I am. All of this, I believe, will have an impact. So all of that will essentially go into the input of the machine learning device. If I have annotations, then I will use those. Annotations could simply mean that is a binding site, or could mean that this site is important for something, or an annotation could be another prediction. Okay. Now, then again, we have the output that is effect or neutral, and there's a certain score that goes from -100. So there are different ways I can translate this output here into integers. such that this unit is between 0 and 100, and this unit is between 0 and 100, and then I can have a final score that in fact this is sort of negative and this is positive. Very neutral then means no effect, very positive means effect. Okay, the one thing that I forgot to put on this slide, an important aspect here, and we can show that here, whenever I look at the feature, so for instance, is this central proline in my prediction that is changed to a y, does that change to a y have an effect? Then I want to know whether it's in a helix or not. But actually what I really want to know is whether that transition, p to y, changes the secondary structure, right? Or changes the degree to which it's probable to be on the inside of a protein or on the outside. So I'm now going to look not only at features, such as secondary structure, hydrophobicity, but change in the feature. How much does this variant change features? That makes it slightly more complicated, because you simply blow up your input space, okay? But other than that, it's straightforward machine learning, and the question becomes how well does it do? You get an answer here in a two-state accuracy. Two-state means you simply count all the variants for which you have an experimental value, whether they have an effect or not. Are you right or wrong? Okay. Those are a couple of different methods: SIFT, polyfen, SNAP, SNP-S3D, and this is SNAP annotated. That is only valid if you have annotations, and the last one, the SNP-S3D, is only valid if you really have structures. And then there's a cool two-state performance, and you see that this machine learning device somehow is performing very well in this environment. You also see these are accuracy versus coverage curves. Accuracy shown here, coverage here, for neutral, for non-neutral, and for buried, intermediate, and exposed. So it performs best for residues in the inside. Okay? For neutrals, and this is flipped for effect variance. Effect variants are predicted best on the outside. Okay, now there is this score that we get. Again, so I should have put this here in terms of 100 and minus 100. I express it again as a probability here, and whether the prediction is 0.8 and 0.2. In this particular case, in the full stop. Or the prediction is 0.5 and 0.51 and 0.49. In both cases, this unit is higher. In both cases, it would predict that it is an effect. But this prediction obviously is stronger than that prediction, right? And this strength of the prediction I want to use as well. I use that as a reliability index. And the reliability index scales such that higher numbers on that side of the curve are more reliable. Lower numbers are less reliable. It's shown as a cumulative percentage that simply answers the question, how well do I do for effect predictions here in red if I force the system to predict for every single residue? That's the answer. Now if I just look at the strongest predictions without knowing anything about the prediction, just the ones that are in this, that are sort of on this side, right? I only look at the ones that have a difference of a certain number, then say that is this set here, and you see for those the prediction accuracy goes from 76 to roughly 86 or 87. Okay, so just looking at the strength of the prediction, it allows you to zoom into the most reliable predictions without knowing anything. I mean, anybody who applies the prediction method can do that. It's trivial in principle, but whenever you develop a machine learning device, the strength of the prediction has an impact, but you have to somehow gauge this or normalize that. Users have to be able to see this curve. Users have to be able to decide, okay, if I picked, maybe a user will use that device only for one particular variant. Okay, and then they need to know, for my variant, the reliability index is 4. If my reliability index is 4, then that is what I expect if it is an effect, and that is what I expect if it's a neutral. Okay? It's just an additional step you have to do. Clear? It's a very clear and simple thing, and in fact that is one of the aspects that makes machine learning more powerful, but unfortunately most of the time people don't use that simple step. They don't provide that to the user, and essentially without that often the predictions are not that useful. Okay, now I can sort of classify and ask, this is now projected again 100, very strong effect, minus 100, very strongly predictably neutral. Can look at a bunch of variants and can ask, well, I know the stronger the effect, the more reliable my prediction. That's what I showed you before. But does my reliability also correlate with the strength of the effect? So very severe effects are in red, very non-severe or very strong neutrals are in green, and something in between is blue. And you clearly see that these three data sets are separated in this score. The reds are much higher here, the greens are much higher there, and the blue is clearly in between. So the blue I can somehow capture for some of those that they have an effect, but my prediction is much stronger here. So that implies it's not, maybe it appears to be trivial, but there are two things that correlate. The more I'm here, the more reliable my prediction, and that also means that I actually predict a stronger effect. It's not a big surprise that stronger effects are imprinted into the system more strongly, but it's not trivial. And again, this is something that you have to establish and normalize. Again, let's look at comparison of SNAP, and that is the first generation of SNAP. So one way in which Jana looked at this was this one here, where SIFT, polyphen, and SNAP, that again is this Q3. Now, when you look at this curve, so this is not, what Jana did here is not 6 months of a master thesis. This is 7 years of your life, okay? After 7 years of your life, you get this. Okay, so this is higher than green and blue, okay? But then comes this sort of sobering part. Maybe you can look at the background of random and say, okay, but you know, actually that doesn't really help you. The fact remains that there is not, right? You see my point. Now she actually had a great idea to sort of justify her happiness. And that was, she did the following. She asked, okay, we see that SNAP is slightly better than polyphen, is slightly better than SIFT. What about the variance for which SIFT and polyphen essentially say the same thing? Let's call them simple, like everybody gets them, okay? They're clearly an evolutionary signal, and the evolutionary signal is found by a single parameter rather than the intelligent polyphen way of doing it. So let's consider that a very simple prediction. Let's now look at the data set of things that are not simple, where in fact polyphen and SIFT disagree. Okay? How well do we do on those? So for those, SIFT and polyphen perform less well than random. This is unfair, of course, because I throw everything away where both of them work, and then I ask how well do they do. So now it's sub-random. Okay? By the way, you have to understand that this does not mean that you could just, whenever polyphen and SIFT don't agree, just use random. This is because finding the way I do it here, I know the result. Doing that up front is not necessarily as accurate. But in this particular case, it looked like that. And now you actually have a big jump, okay? So this, you may argue, whenever, so there were two methods before I started 7 years ago, but whenever they agree, and that's the case for many cases, or for some cases, whenever they don't agree, that's the case for many important cases, then in fact, my method is much, much better. Then we get into the development of SNAP2. Most of that was done by Max Necht, in our group, who just finished his PhD. And the idea really is, first, there's so much more data that has been added. One set of data comes from disease variants. So where we know that a single variant of it causes a disease. SNAP1 never really used that. SNAP1 used typically some of this, but on a relatively small scale. In fact, Max could add 23,000 variants right away to the 38 that he originally had to bring the totals to these numbers. He brought in some new imbalance because, again, the neutral, despite the enzymes here, were smaller because he did not do an effort to increase that set. We could have done that. But we essentially believed initially in a quick step where we simply use those data and then we see. And then the argument was, then we have some additional data set that is not contained in any of these, where we can assess the method. The next argument was that, so we have a larger data set, do it again, machine learning, right? The next argument was we have some pieces of information that we have never put in the original data set. Some of it is biophysical features, some of it relates to contacts between residues. We could gauge and sort of use the alignment information in a slightly better way and possibly improve by putting additional information in there. You see that, you may remember that this set here has increased of important input features. And then there are, in fact, two new concepts here. One is, let's just train a machine learning device that doesn't use information about alignments at all. We know it's going to be worse. But by differentially looking at one that uses alignment and not, we somehow get a mechanical reason or causative reason why we believe this has an impact. So if the machine without the alignment information predicts equally strong as the one with alignment information, we absolutely know that this is an effect upon structure. Or we know it's an effect upon some part of this machine here that uses no alignment. Okay? And that is may be an important thing to know. And then we use one that is never trained on disease data. The SNAP2 in these curves effect neutral, left neutral, right effect. In fact, you see the shift in overall performance. The dark blue is SNAP2, SNAP1, SIFT, and random is down here. And that, in fact, is the one without alignment. So the one that does not use alignment is clearly worse than the simplest one parameter SIFT solution. Okay? It's not by no means competitive. But in cases where you have poor alignments, this is important. Again, what is relevant here is the differential way of looking at it. And the dark blue is higher than the light blue. Now when we put polyphen into the equation, again, the effect neutral in an accuracy versus coverage plot, polyphen is the dashed here. Then one thing is that the dark blue is above everybody else, but we would have liked to be have a little bit more room there. So the many things that Max put in brought surprisingly small of a difference. Nevertheless, one way in which people typically combine methods is by saying, well, I accept the prediction if and only if two methods have agreed on that prediction. So if SIFT and polyphen say that's an effect, I call it an effect. Whenever they disagree, I ignore the prediction. That's what many people do in the way they apply prediction methods. If you did that in a naive way, then you would reach that point. And you see that the blue of SNAP is clearly above that point. So it's not a clever strategy, is what this one shows. Again, the difference between this point and the blue point here is not very high, but it's significant. It's a large data set. So the error bars are invisible. Not because they're they haven't been done, but they essentially is the line width, if you want. That's the error bar here. Okay, here's the story that I showed you before. In this particular case, it's for human. And so we essentially look at tough cases, at cases where methods don't agree. And for the cases where methods don't agree, SNAP2 is really more often, is typically very, very better than everybody else. The slides that I showed you before where the light blue was better than polyphen and SIFT that is the polyphen 2 version, and I previously showed you the polyphen 1 version. in fact, for this data set that Max used here, now SIFT for the difficult cases is still better than random. That's also slightly different. But good news is SNAP does outperform the others. Again, it works very well in looking at the reliability index. Now let's go beyond single predictions. So what I mean is simply that what I predict so far is the effect of one change. The entire sequence, one amino acid that changes, and I predict the effect. What else could I try to predict? Okay. What else could I want? May I want to know?
The combination of different.
Yes, that would be a great thing. So when I have briefly spoken about, it could be that we have a variant here and a variant here, and that something here changes from a small to a large residue, and something here from a large to a small, because they are related in space. So if they are inside of a protein, by replacing the large residue, the small residue by large residue, you sort of push things out, but you could compensate for that by creating space, by having a compensating variant. Now, yeah?
But aren't you already taking that into consideration when you consider features of a protein, for example, the 3D shape, two-dimensional shape?
Well, so far, implicitly as a statistical average, I do. But explicitly for that particular point, I do not explicitly ask, these two, could they co-occur? Are these two things that could compensate for each other? Is that a disease that I could recover by that variant? That is what we try to do, and there's a method of doing that. I will not get into that today. Anything else that we may want to look at? Well, one idea here really is, since I can predict the effect of a single variant, I can look at every single position in the protein and vary that to, say, alanine. That's what people do experimentally. Or go a step beyond that and, in fact, vary it to all 19 non-native. What if I change that to any other letter that could be at that point? Okay? And maybe that would relate to some hotspots. Maybe at least there are hotspots of change. So maybe there are places where I change against anything it has an effect, and others where I change against anything I don't see an effect. So I sort of have to come up with a method that uses that idea. Simply, you try to mutate against different residues and see the effect. And again, you simply, so in principle it's the same thing as before. You initially predicted 1, now you do all 19, and you somehow find a way to average over it. When you average over it, then you can simply compile all the possible effects and compile the correlation of a particular variant. So all the possible effects at one particular position, how does that correlate with mutating it to alanine? How does that correlate with mutating it to cysteine? For each of these, you can effectively measure with the prediction probability of a correlation. The correlation is highest for alanine. Anybody has any idea what that means or why that could be the case? So essentially, yeah?
Correlation for a change. So if I change whichever residue with alanine, it has a high effect.
It's the most representative effect. That's what the correlation here is a correlation with respect to all the possible changes.
I don't understand that.
So I'm looking at one particular position, I change it against alanine, okay? And against an A. And then I do that for all the positions in the protein. I have a protein of 100 residues, I do 100 2A variants and predict the effect. Now I do 100C, blah, 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 okay? Well, not 100. 100 minus the ones where the C is already there. And now I compile all these changes. Say 100 times 20, roughly. 100 times 20 minus 100. So 100 times 19. I have 1,900 possible changes in my 100 residue protein. And I compile the average of 1,900. And now I ask, at every single position, what's the effect for the 19? And what's the effect of the alanine? How does that correlate with the effect that I predict for all? Okay? If the value is 1, then alanine just looks like everything. It's completely representative of all changes. If the effect is very low, here for the D, then the change to D differs very much from all other variants. Okay? Is that clear?
So changing D is a high effect.
No, changing D is very different from all other changes. So what I look at here is really, let me turn it around.
Sorry.
I'm looking at the question, if I, experimentally what people do is they take a residue and typically change it against alanine. And then they observe an effect. And they do that historically because, on the one hand, you believe that alanine is relatively neutral. On the other hand, historically, at some point it was easy to do that. And that is why alanine scans became a word for simply testing the effect of variance. And that has been used initially to test where functional sites are. If I believe, so I have my protein here, that binds, that's a binding event, and I don't quite know where the binding site is. So I pick this residue here, and I believe this is a binding site, and I pick that residue here. That may be a binding site. Now if I mutate that one here, I expect no effect, expect some effect on the binding. If I mutate that one here to alanine, I expect no effect. And ultimately, experimentally, this is one means by which people detected binding sites. By simply seeing that the change to alanine had an effect or not. It's not direct because the effect could be, so the protein could come back here and you could change this alanine that would influence this one here. Although this one doesn't touch, you could have an indirect effect. And that is always a problem for the alanine scans. But historically, this is the way people did it. And now, what I'm trying to do here in silico is simply ask, how representative is the change to alanine? Okay? And since alanine is by far the highest value, I make the statement, yes, experimentally changing to alanine is, if you want to choose one, then choosing alanine is the best to do. If you chose one, or if you chose a variant, the worst that you can do is essentially the Y, the tryptophan, the Q, the negative charges, and the functional residues, the polar residues here. That's what you see. It's no biggie, because you completely understand that biophysically, why that is the case. But it's remarkable that now you just have the same method that predicts the individual effect of a single variant, and you run it again and again. You essentially do a recursive run over your method, and you get out some truth or some observation that is compatible with biophysics. Again, it will not surprise any physicist is not convening any value, any insight, but it is telling you that your method does something. You recover information that you did not put into the system at all. The system never learned that. At least not by you, not explicitly. So this in silico, comprehensive in silico variation or mutation does work. That's what you learn from this. It's a consistency point. Here, what we look at, or what Jana looked at, is she looked at this particular protein, and in this particular protein she asked the average effect of variant. And she saw that there's a region here where essentially everything you do has an effect. And then you see many regions where that is very different. And that region is in fact exactly the functional binding site of p53, one of the most important transcription factors related to cancer and many other diseases. So you have a simple tool that tells you immediately something about the overall, let's call it structure, or the overall functional importance, which you never put in. Okay, that is not a method that predicts hotspots, that predicts binding sites, that predicts domains, or that predicts regions. But in this plot you immediately see it falls out right, clearly there. We can now use that, and I'm zipping forward, to predict binding sites, binding hotspots, and it somehow works. Here is an example where the two binding sites, and the slide is not the highest quality, the redder, so you somehow see red here and blue. The more red you see, the more of an effect there is. And if I could rotate that, you would see in the movie that somehow is not in here, no, it's not in here, you would see that essentially the more red is exactly in the binding sites. So the regions where you have more effect predicted for any variant, any of the 19 non-native, is the one where in fact you have the binding site. Here is the same seen by changing the red according to some weight, and the binding site comes out in a clearest way in a particular scoring scheme. Now you can then apply that method again here in this particular case for a particular situation. That's a melanocortin receptor. It's one particular protein that has 7 transmembrane helices, 1, 2, 3, and so forth. And you can simply ask, what are the regions that are predicted to have an effect? Now this was an important target for us because the method had never been developed on membrane proteins. So essentially the question is, does it also work on those? And we labeled, Jana labeled the ones that are predicted with a strong effect. Again, all where everything that you vary to would have an effect. We put in simply the ones that are experimentally characterized, and we found points, or we found situations where there was no experiment implying that particular position in a strong effect, and we had predicted one that was a very strong effect. And again, people began to experimentally work then on these predictions. A very successful story. But in some sense, the most remarkable example here was we had cases, we have this, in fact, there's 2 different receptors, melanocortin-like, they are 50% sequence identical, and there are some residues in this one here where the position, here's an example. So, no, that's not an example. Wait a minute, here's an example. The phenylalanine. So we have some positions where both of them have the identical letter, yet we predict a different impact of the variant. So playing our 19 non-native game on this one and playing it on this one gives a different result. So we have 2 proteins that behave in the space of possible mutations differently, and we can see differences at places where they have the same amino acid. And that is remarkable, because there's no other method that can do that. When you have the same, so you have an alignment of 2 proteins that do the same thing, you have positions where they have the same residue, that is the same, right? Overall is the same, locally is the same, no difference. And suddenly we have a method that can still see differences. And the differences it sees essentially are the susceptibility to change. How susceptible is that position in its local environment to change?
How?
Ultimately, it comes from the fact that the position here is in a slightly different structural environment. Again, it's a predicted average in this particular case, but you see an environment, a sequence environment and a structural environment, and you see both effects. And there are cases where the identical residue has the same structural environment and everything is the same, but there are cases where it's not, and the method can detect that. Okay, that's the remarkable aspect of this one. Then now we can look at this heat map. So here runs the sequence. This is the effect of the 19 non-native. Red is more effect. You see there are regions that are sort of overly green. There are regions that are largely red. Red is dominated in this particular protein. Now you can again, that's what I meant. So there are regions where sort of everything has an effect. Regions that are sort of everything doesn't have an effect. Now we can essentially look at particular sites and we can ask, okay, do we have cases where in fact everything has an effect, but we are we talk about a position where, for instance, we have very hydrophobic residue to hydrophobic residue. If we looked at Blossom, if we looked at the biophysics, we would call hydrophobic to hydrophobic neutral. Nevertheless, I predict an effect. Okay, let's look at the ones that are non-trivial, that are not expected, where the effect is predicted, although we would expect it to be neutral according to Blossom. Okay, then let's look at the ones for which I have a predicted correlation of a contact. So first of all, that's the first filter. We have take all that is red, we filter out what is sort of not trivial. Let's call it like that. Then we look at things that where contacts or correlations are predicted. And then we essentially zoom into, by these two filters, we zoom essentially exactly into the binding sites. So we begin with something that is overly red and really not helpful. First filter is trivial, second filter is not that trivial, but again it's something that is available for many proteins. And we get down to a level where for this particular example we tried one, we get very, very close. In this particular case we hit the binding sites. So what this is supposed to show you is that we begin with a simple method that predicts an effect of one residue. We expand it into something else, heat map prediction. This heat map is too red, so we find a simple filter of the heat map and we get to a functional point. Some of these steps here are not automated fully. So there is still some improvement to be done. Ultimately, at this point, that works in the hand of an expert. Okay, where can we go from here? One way is to look at the changes upon structure, the dynamic changes, and that's something Mark Offman worked on. Here are two cases where that led to some statements about diseases and about development of drugs. But those are sort of projects where you use now method with expertise to model something. Okay, those are detailed projects. Those are not thorough methods. And here's an example why this is complicated for these individual predictions to come up with thorough methods. This is a set of SNPs Neanderthal. So Svante Pavo's group in Leipzig, Svante Pavo and Janet Kelso, essentially got the first sequence of Neanderthal. And when they looked at this first sequence of Neanderthal, they came up with 78 non-synonymous SNPs that have the following. They change the amino acids. We're talking about coding variants that change the amino acid and that are such that chimp and Neanderthal have the same amino acid there, and no human has that amino acid. Okay? And of all of those, in the entire genome, 78 was the answer at that point. It's an incomplete genome, and the number is higher now. But that is the ballpark figure. So I believe at this point maybe they have 200. I can't remember what the latest number is. But it's still a relatively limited number. And again, this is important in the background. Every pair in this room has 20,000 differences. Okay? Even if you brought that up to the level of 1,000, we're still far away from that number. Okay? But again, so there are additional constraints. It's not because we are more different from each other than from Neanderthal. Let's not jump to conclusions here. It's simply, I'm only bringing these numbers to make the point we want to discover so differentially studying, or they want to discover by differentially studying human and Neanderthal what makes the difference. It's very clear. That's what Svante Pavo sets out to do genetically, to describe how apes developed, how human developed, where we are coming from. Okay, so one thing that we can simply do is, in our group, Schuyler and Yannick have worked on that, we can ask what's the impact of these 78. For instance, what's the impact on in the prediction that we have on this 19 non-native? And one other way that Arthur has worked on, when we look at them, how the proteins that have these 78 what can we learn about their protein interaction networks? And so I'm only going to show you the part here that didn't work, so I'll show you only the negative data today, because for this story there is no positive data. So what you see here, for the 78 are all the protein interactions they have. So all the interactions between the proteins that carry these 78. Okay? And you see this is a very rich, dense network of zero connections. It's just nothing known. So not a single one of these has any interaction to any other of these 78. So these 78 are not connected to each other in terms of so they all sit on proteins that do not interact. This is clearly surprising. In fact, I can't remember what the number of protein is, but Arthur, wait a minute, he had only 69 of these 27 mapped to the network. But he had this point that, first of all, I believe this was, for him, was a surprise that so few mapped. But then that none of them, whenever he came in the past with data sets that somebody had given to him, there were more connections to data sets of in the ballpark of 30 or 20. More of those were interconnected. So he was surprised by that. He still saw some other effects, but let's not get into that. When we essentially want to study the effects of the variance in us in order to study disease, there are additional problems, and one of these additional problems has to do with making all the sort of annotations available. Protein is one way in which we are doing that, and I believe I ultimately also want to bring this slide up. So thanks to Laszlo Kajan and Dayak Duff, Protein runs since '92. Is the first internet web server in molecular biology on the internet. So this is long before Google for some of you. And it has been not easy to keep this up and running for so long. Okay, now let's just look at a few data sets and the impact, or the prediction of impact. And what we show here is the SNAP score of severity. 100 is predicted to be very strongly effect. Minus 100 strongly predicted to be neutral. It's a cumulative plot, so I'm asking what fraction of my data is above a certain threshold. It's predicted with an effect stronger than some number. Here, stronger than 50. The data sets that I show you here are disease-related, black. So those are all the residues that are known that cause a particular disease, where one change causes a disease. That's the effect prediction. Red is my training set. Okay? Have we discussed this story before? Yeah. Yeah. And the other side, this is random. The other side of the equation I don't have here. But let's go into another one. So in the other one, this is actually the same thing. Here it's just a slightly different data set. This, by the way, is SIFT. The reason why yellow is higher is because SIFT overpricks. So the entire is also much higher than in this particular case is SNAP2. That's training. SNAP2 OMIM, and this is SIFT. But again, for neutral, SIFT is also above. So it's an overprediction. Here, green and red are, one is for single residue that affect the disease in people is red, and this one here is in animals. So the disease in animals has even a stronger effect. Any idea why that could be the case?
Because we can study them in a shorter cycle. You have more experimental data.
No, actually, the data here is much less. That I would assume that you are right, but the data effectively available for us is less. And ultimately, that's a question of curation. This data is more easily made available because there's a higher pressure to make that data available, I guess. But why, if we believe, so yes, there are error curves, but we do believe that the green is above the red. So disease in animal have more effect than diseases in human. Why could that be the case? It's a very simple, I assume it's a very simple explanation. So let's get back to the mice. When the mice don't have hair, nobody calls that a disease in an animal. When we don't have hair, this is clearly considered a disease. So the types of things that are called disease in human are just not as severe as the types of things that are called disease in animals. So the green is typically a dead animal, and some of these red ones are still alive. So now we get into another one here that when people mostly do animal model, the human diseases as a model in mouse. So what they do is they take the variant in the human protein. So one way of doing a mouse model for a human disease is to really take the entire human protein and put that into mouse. Another way of doing that is essentially you have a variant that you know causes a disease in a protein that the mouse also has. So you simply put that variant into the mouse and see what is happening to that mouse and study that in a molecular way. Now, we can do that in silico as well, right? We can take the mouse sequence and put in the human variant. My question to you, if I did that, would the effect be stronger on the human protein? So I have the human protein here. I have the variant of the human protein at this particular variant, and say that was an E to K. And then I have the mouse, and I do the mouse protein with a variant, so I put a K in here. And the question is the mouse to K, does that have a higher predicted effect than the human to K? So we expected.
If it's something related to, for example, digestion, if the mouse doesn't eat certain things that we do, and that infers that we are more likely to die of a certain disease because we eat that food and don't digest it the right way.
You're right. You're right. But again, so I'm asking now. I believe we had about 120 variants here, and they come from different diseases. And most of them are pretty severe diseases. Okay. So now, sorry, I'm actually doing the other way around here. I take all human diseases. So that's 20K. 20K variants. I put them into the mouse sequence, and I predict the effect. Do I have a higher effect or lower? Most of the time, actually, the effect is lower. The light blue, that's the human disease or the human variant put into mouse, and that is below the red, which for us was a big surprise. Which essentially means that things that are completely independent about the situation of the mouse, maybe has a different immune system, and may have a different way to handle certain things. But just on the level of the sequence alone, the simple fact that you take a variant from one into the other, in fact, reduces the effect simply because you have a lower space of reaction. Well, I don't know why, because. So we have an emotional explanation why. We write a whole paragraph what we believe is the reason, but actually we don't know at this point. This is an observation. And this is something that is important, again, where you develop a simple prediction method that tells people who work on mice models, be careful. That's all it says.
What do you use to predict the light blue line? Did the SNAP learned on human?
Well, actually, SNAP is not learned on human. Most of the variants that are used in SNAP are not from human. Most of the SNAP data is not from human. In fact, we used the one that did not use disease on this one here. So it's 38, of which most are not human. But that, yes, we used the standard SNAP. Not trained on disease. Okay? So let me tell you the story without the slide. The slide is complicated again. Now what I can do is I can take the variant that causes the disease. I know they have a strong effect. And I know that the conservation of that residue is an important signal. So what I can do is I can find all the other residues in that particular protein that have a similar level of conservation and mutate against that. Or I can ask, this is one way of asking, and then the question becomes, is the OMIM variant, is the one that causes a disease, standing out in terms of conservation? And it turns out that the variants that we see that cause disease are always in a particular protein the ones that are most conserved in some sense. Not the ones that have the highest actual value of conservation. But it's typically such that we hit the positions that for which mutations to anything have a big effect. That's what, if you have enough time, this slide shows. Which you may argue is expected, but for us was in fact the strength of the effect that we saw was a big surprise. Okay. Sorry, I tried to do too many things in today's lecture. Thanks to many, many, many people. I just put up some faces. Today I briefly talked about Thomas, a lot about Max, with some of Arthur. I briefly talked about Tanya's. Laszlo has the protein server running. Guy Yakdaf is here, another one who kept the server running. Annotations, Andrea Schafferhans. I did not talk about the method that Christian Schäfer developed. Christian Schäfer, what I talked about today was the impact on function. Christian Schäfer worked on methods that predict the impact on structure. And the reason why I didn't show you is because ultimately the tool is published but not really available, because I believe Christian didn't believe enough in the usefulness of his tool. And in some sense the message here is believe more in what you do, in some sense maybe, because I believe it would make a difference to us if we had it. And with that, Yannick, I showed data from Yannick, who's now doing his PhD at Rutgers. And I showed data that Jonas just has compiled on the Omiya story. Thanks for your attention. And from now on we go into